Welcome to PMAC Interview. I'm Pavit Pinvijit, and with me is Professor Peter Cullis. You started out in physics. Why did you change your biochemistry?
It was somewhere partway through my PhD, I realized this is not the way I wanted to spend the rest of my life. Physics is it's a tough discipline, and a lot of the important discoveries in physics have been made a long time ago, and so the theories are very well established. It was very difficult to discover something new, and as an experimentalist, because I did experiments, there's a hierarchy in physics. There's theoreticians and then there's experimentalists. And as an experimentalist, if I did an experiment and I found something that didn't agree with theory, it was very likely that my experiment was wrong, not that the theory was wrong. So I found that dissatisfying and decided that I had to do something else. And I was very lucky because I got a fellowship to go to Oxford University and into biochemistry, which is what I wanted. I wanted to get into the life sciences because I could see that there was a lot of problems there and looked very interesting. And I could use some of the techniques that I developed or that I knew about in physics to study biological systems. So that's what I did.
So what do you work on when you first started in the field of biochemistry?
I first worked on building an NMR machine, which is a very complicated machine. I got to Oxford, and they had an NMR machine that, they had the magnet, which is an important part, but nothing. And so we had to build it. So luckily, I had a physics background and knew what to do. But no, the first thing I started to work on was lipids and membranes. And I had the good fortune to meet a postdoc who came from Holland. Who was was an authority on lipids, on membrane lipids. And so we teamed up together using NMR to study the properties of these lipids. And that was really fascinating. I got more interested in the lipids than I was in NMR. So, everything went from there.
So the lipid nanoparticle technology has been your thing for how many years now?
Well, since about 1975, I guess, 1980 in particular. But one of the things biological membranes are very complicated, and so in order to study them, you have to make simple systems with some of the components to find out what their properties are, and they're called model membranes. And it turned out that these model membranes, in addition to revealing how membranes worked, we found we could make little nanoparticles that we could load with drugs. And so that's what got us started about 1980, 1985, somewhere in there. We found we could get cancer drugs packed into these nanoparticles, and that they potentially had uses, as a delivery system to get cancer drugs much more specifically to a tumor, say, as opposed to other places where they have nasty effects. This is still something that's going on in my laboratory. What we can get them circulating around the body. Now what we're trying to do is to blow them up in the region of a tumor to get much more cancer drug at the tumor site and not in other places.
Have you ever thought of using LNP as a delivery for vaccines?
That was not the area, the direction we went in or were going in at all. We started off. We worked with the cancer drugs, which were very small molecules, and then about 1995, we thought, gee, well, maybe we can deliver some of the larger ones. Such as the RNA or mRNA, or a small interfering RNA is another form of RNA. And so we teamed up, we first of all had to develop systems that were not toxic. One of the things that we learned with our work with cancer drugs was if you have systems that are toxic, that have nasty side effects, you have a great deal of difficulty getting them into clinical trials, and that always is a problem. And so one of the things which we tried to design was a carrier system that had really reduced toxicity. And so we found a particular kind of lipid, it's termed an ionizable cationic lipid, that had this property that we could load it at, say, a low pH. And the low pH, if you have a lemon, inside a lemon, it's kind of acid. It's a little, anyway, so if you could load them under that condition with, under pH 4 is what is termed, then we could bring it up to physiological pH, and they were not charged anymore, and so therefore not toxic. So once we did that, we had a way of loading the systems with really anything, any nucleic acid, RNA or DNA. So then we associated with a company in Boston called Alnylam. They had a piece of RNA that they wanted to deliver to the liver, and so we worked with them for 7 years, and we gradually got systems that were better and better. And in 2012, the formulation that we developed went into the clinic, and then to be tested to see whether or not it was going to work against it's an amyloidosis problem that or a protein that's produced in the liver. If there's any mutations there, it can cause problems. So then at that point, this is actually mainly in a company that I started called Acuitas, we said, okay, if we can deliver this small interfering RNA to the liver, and that was basically to shut down production of a gene, maybe we can deliver messenger RNA to produce a protein. And so that's what we did from 2012 onwards, and we were just really getting better and better at producing proteins in the liver, because there's lots of applications for that. When we got called up by Drew Weissman and Katie Carrico saying, we'd like to try your systems as vaccines, when instead of going in intravenously, going in intramuscularly, and to see if there's a good immune response. And it was that was really good luck. Really, it turned out to work very well. But we weren't going in that direction at all.
So it's quite fortunate.
Absolutely.
Yeah, absolutely. Well, you have so many patents under your name, and you have founded so many companies. And how do you get all this business savvy, doing all this business funding and starting ups?
Yeah, it's. You learn the hard way. The first company we started off was about 1985 or something like that. We developed an instrument in the lab that was very good for making lipid nanoparticles, and so I gave a talk. I think it was in New York. And somebody in the audience said, I want one of those. So I said, okay. We had it made by a machinist in Vancouver, and so I put him in contact with the machinist, and then 2 months later I got this call saying, well, I got this thing, but it doesn't work. And of course, the machinist had no idea what it was being used for, so there was no quality control. It didn't make sure that it could actually do what he bought it for. And so at that point we said, okay, well maybe we'll set up a company where we make sure we do all the testing and make sure it actually works. So that was the start, and we knew that companies had to have a what do they call it, a statement, that's a motto for the company, and so we thought, okay, well this is me and 4 postdoctoral students in the lab. So we thought, what do we really want? Okay, we want free beer every Friday, and
sounds nice.
And parties whenever possible. So that was our slogan for the company was free beer every Friday and parties whenever possible. And nobody has ever paid for a beer in my lab since. So that's one of my proudest achievements.
Nice.
And that started us off starting companies. That from then on, it was a good way. It turned out to be a very good way of keeping a good team together, because I had a very at that time a very accomplished team, and I couldn't. It's hard to get that set up in a permanent way within a university, and so I found if I started a company, then I could keep that team together, and we could then. And that was really it really helped. Without that, we wouldn't have achieved what we achieved.
Great. So as a private and public corporation, I think.
Yes. Yeah. We had one company we took public, raised money on the stock exchange. Other companies remain private, but yeah.
Excellent. Well, you frequently talk about personalized medicine. Tell us some more about that.
Oh yeah, this is a bit different than the lipid nanoparticle work, but we have a problem in medicine now that it's a kind of a one-size-fits-all approach. which is, there's good reason for that. But all the drugs that we develop, are really meant for,
The mass.
the mass, the whole, all people. And it means that, there's, there are people that don't respond that well to those particular drugs. And so we see this problem all the time. And so it's really meant that the development of medicines has been, is really very difficult and very expensive. And almost invariably, you can develop a new drug and there'll be somebody who will have a bad reaction to it. And so we know what we need to do is to get medicine that's much more focused on the individual, so that we just give that person the medicines that work for them, and they may not work for other people, but they, so in order to do that, we need to know much more about the individual. And now we can do that because we can do the genomics, we can sequence the genome, we can measure the proteins in the blood, we can measure metabolites in the blood, we can look at the microbiome, which is all the bugs that are living in you and on you. And from there you can get a data cloud which describes an individual very specifically. And so it's and then you can reflect that against, say, the world's clinical and scientific literature and say, okay, what diseases are you trending towards, but more specifically, what drugs will work for you for a particular condition. Now, it does coincide with the mRNA and the gene therapy approach, because we're now going to be moving into an era where we get not just personalized medicine in terms of drugs that will work for you, but also we'll get personalized medicines that are made for you. So that for example, if a child is born and they're not producing a protein that is vital to life, it might be a clotting protein or many other, some of these lipid storage diseases, etc., that could be lethal within a very short time. We can now, once we find out what the problem is, say, okay, let's get the liver to make that protein. And so we can, once we know what the protein we want, we can make the messenger RNA, package that up with a lipid nanoparticle, and then inject it, and you'll have the livers will start to make it, and they make that protein. So I see this as a huge, we have very personalized medicines that are highly targeted for individuals. It's a very different kind of medicine than we're used to, because you can do all of this in a matter of a couple of months, as opposed to waiting for 10 years and spending $1 billion. You can, developing these medicines is much more straightforward. From a regulatory point of view, and this is not the usual way that medicines are made, but I think it's going to cause a revolution in the medical way the way that we administer medicines, or way that we treat patients. So it's going to. You can imagine this is. This is an area where there's going to be a lot of people demanding, if once they know that this is possible, you can see that there's going to be a huge demand if they have a child that's suffering, or increasingly for older people too. There's a real need for much more personalized approaches. Our treatment of older people right now is we give them more and more drugs, and this is not a good idea. Yeah. We need to be able to say, okay, how do we treat some of the diseases of old age in a much more specific way? Everything from Alzheimer's through to to rheumatism or arthritis. I mean, all these things are and we can do that now with these new medicines.
Okay, well, exciting futures. Do you have any advice for the next generation of scientists and doctors who might lead a life of?
Well, yeah, I do actually, and it's just to be aware that we're entering a new phase of medicine. That we've gone through one revolution, which is the development of small molecule drugs. I mean, everything from the antibiotics to cancer drugs, etc. And then we've gone through another revolution, which is the biologics, some proteins that are therapeutics, monoclonal antibodies and things like that. And now this is this third generation of drugs, which are the nucleic acid-based drugs, or the gene therapies. I think you're going to really revolutionize almost any disease you care to name. You can figure out an approach using these sorts of gene therapies to treat it. So I would say for anybody right now that's contemplating a career in science or life sciences or in medicine, this would be a great area to go into. I mean, it doesn't get any better, right? I mean, you have the tools, but it's still pretty much an unexplored. You know, the applications are not really worked out yet. So I think it's going to be a very exciting time.
Oh, excellent. Well, thank you for talking to us today. I hope to talk to you again soon.
Okay, it's a pleasure.
Well,
thank you.
And that was Peter Cullis, the recipient of the 2021 PMAC Award. You're watching PMAC Interview. Stay tuned, and please don't forget to subscribe to our YouTube channel.